this is Martin Wilson welcoming you to this Bite-Size Bio web seminar. Today's presentation is titled Basic Methods for Immunohistochemistry and is brought to you today by Mike Millar. Mike is the Facility Manager for Immunos and Imaging at the Shared University Research Facility, or SURF, at the University of Edinburgh. During the presentation, please type any questions you have in the questions box on the right-hand side of your screen, and we will put these to Mike at the end. So now, over to Mike Millar for his presentation.
Okay, well, welcome everyone, and I'll apologise now for my rather broad Scottish accent that I have here. If any of you have problem understanding me, just take some questions. Martin. So what I'd like to cover today is really the approach that we take to delivering immunohistochemistry and immunofluorescence staining to a wide research community, and that's primarily kind of academics throughout the University of Edinburgh and the wider research community. I'm supported by a great bunch of people, so I have Mel, Bruce, Debbie, Laura, Robin, Gary and Mariana in the lab with me, and they all help me to deliver these services. Now, if you need further information about what we do, it's available on our website. We're currently in the process of revamping this website as we're aiming for GCRP accreditation in the coming year. So we're hoping there'll be lots of protocols and SOPs posted here in the coming months. So what I'd like to do is just cover our approach to immunohistochemistry and the reasons that for taking the approach that we took, and what technologies we have used, and currently why we think polymer and tyramide-based detections meet our current needs. And if there's time, I'll maybe touch on some future directions. Now, our strategy from the outset was to aim for the best morphological preservation possible, even if this risked sensitivity. And that was because, certainly initially in my sort of research career, we wanted to conduct a really sort of detailed morphological analysis on the samples that we were looking at. So we made preservation one of our top priorities, which is why we went for paraffin-embedded samples or resin-embedded samples over frozen sections, for example. But we also wanted our tissues to be suitable for looking at messenger RNA expression by in situ hybridization, protein expression using immunohistochemistry and immunofluorescence, and other tissue molecular probing techniques such as tunnel paraptosis. What is probably more of a priority for me now is that we can deliver techniques as easily as possible, because we do have a lot of different customers working on a lot of different types of tissue from a lot of different species. So ease of delivery is now probably one of my kind of most important targets. So this is kind of the one and only science slide that I'm going to show. The rest will all be sort of technical about our approach to staining. But it's just to explain why we needed to take the approach that we took, which was to focus on preservation. Okay, so if you look at the bottom left panel, that's a cross section of a rat seminiferous tubule that's been stained for a messenger RNA using a non-radioactive in situ detection method. And what we can use is, using this probe, we can identify, or we can see quite clearly, that these seminiferous tubules have different expression patterns. Now, if you look at the panel in the top hand left, what distinguishes these seminiferous tubules from each other is very subtle differences in the morphological characteristics of the cell population there. And that is kind of explained in the sort of large panel on the right hand side. So I think you could probably imagine that to analyse that tissue accurately and get meaningful data out of it, as well as being able to label the tissue, preservation was really, really important. And I think for most of these experiments that you're going to be doing with immunohistochemistry, using antibodies, either using colorimetric detection or fluorescent detection, the end point is going to be some sort of imaging application. It may be straightforward photomicroscopy, it could be confocal microscopy or image analysis procedures. And what we really try and do is to support researchers or deliver techniques hands-on so that people get good, reliable publication quality data. And a lot of the work that I'm presenting today was facilitated by my colleague Sheila McPherson, who's in this panel up in the top left, training a bunch of students. And this kind of sort of ease of use so that people can get results easily is, as I said, a priority for us now. So starting from the kind of least sensitive method in our armory is the direct method. Now, it's a nice straightforward method where you would buy a primary antibody which is already conjugated to some sort of label. It could be either a fluorochrome, gold particle, or an enzyme. And you usually just apply that to your tissue section, which has probably been pre-treated in some way, and it should bind to your target protein. and give you a signal. Now, if you've got an abundant protein and it's well expressed, then this is quite a valid approach. It's nice and easy. What may restrict it is availability of reagents. There tends not to be a huge range of primary antibodies which are already conjugated. And once you buy that antibody that's conjugated, you really are fixed to going with that detection system. So it's not as flexible. One of the potential advantages is, of course, is that it's quite a popular detection system in flow cytometry. So people want to do fluorescent activated cell sorting, for example, and then use the same reagents to detect kind of cells and to detect protein in fixed cells or tissues, then it may be approached. Generally, it's a bit hit and miss whether these reagents are compatible, because generally the antibodies for flow are to detect antigens in their native state, i.e. they've not been fixed in any way, whereas usually in kind of the work that we're doing, where we're looking at fixed cells or fixed tissue sections, the proteins have been changed because of the fixation protocol. So, it's not often compatible there. But even with this kind of most basic technique, you can still play about with it, and it gives you choices for sensitivity, and it's something that probably a lot of people don't consider. So your choice of enzyme could affect the sensitivity of your detection system. And there's various enzymes out there that you can use. There's phosphorylated oxidase, alkaline phosphatase, and glucose oxidase, for example. The type of hapten you use, whether it's gold, biotin, streptavidin, could also have an effect on the sensitivity of your detection. And if you are going for fluorescence, then also the fluorochrome that you choose, the excitation and emission functionality of your imaging system would be important to get the best out of that particular label. And also fluorochromes with different Stokes shifts, so where you have a higher Stokes shift, you'll have the brighter fluorescent signal. So they can all be considered when you're choosing that reagent. And finally, something that, again, a lot of people may not think about is the sensitivity of substrates. Now, we've used several substrates in the past for colorimetric detection. DAB is kind of fairly universally used. MBT is another substrate, which is for alkaline phosphatase, and that was used fairly extensively for non-radioactive in situ. Now, generally, the rule of thumb is that with an HRP and DAB detection, once you put the DAB on, if you don't have a signal after 20 minutes, it's probably not going to either materialize or it's not going to get any stronger. However, MBT and alkaline phosphatase is a combination that allows the colour to develop for a period of up to 48 hours and possibly beyond that. So it was popular for in situ because you had this low abundance target, but you had this detection system that kind of just amplified colour over time, which made it a good and sensitive choice. Now, a much more flexible approach is using something like an indirect method, where you would bind your primary antibody to your target protein in your cell or tissue, and then you detect that with a secondary antibody which has some sort of label on it. And that gives you a lot of flexibility because you can mix and match your reagents. So you could have maybe a panel of 10 or 20 primary antibodies with one or two or more secondaries which can be used to detect that. So you're not fixed to your detection system. There's a bit of application with an indirect method, but it's still not as sensitive as some of the current methods. But again, if you've got a good sort of target protein that's well expressed, then it's still a very kind of valid approach. And I think that sort of detection system is very popular in Western blotting. It's not something we do a lot of personally, but I do see a lot of protocols where the indirect method is probably the method of choice. And there seems to be not a huge uptake that we can see in our institute for people when they're doing Westerns to look at much more sensitive detecting, more sensitive multi-step techniques. So a few years back, we were using sort of label streptavidin biotin methods fairly extensively, and it was really because of the sensitivity that this approach gave us, and also the flexibility. So in this approach, it tends to be a three-step approach where you bind your primary again to your protein, and you detect that primary using biotin-related secondary antibody, and then you would visualize that binding using a labeled streptavidin of some sort. Now, of course, this labeled streptavidin could be any type of streptavidin. It could be any fluorochrome that you can buy. It can be enzymes. It can be gold. It can be lots of different things. So very sensitive technique and very flexible. The main downside for us for using streptavidin-based systems is that quite often the sort of reproductive tissues that we worked in, and certain other tissues like liver and kidney, I believe, do express quite high levels of biotin. And if you don't block that endogenous expression that's in the tissue, you can get a false positive signal. So It's not a disaster because you can do an antibiotic blocking step, you can add that to your protocol. But what it does do is it starts to add time and it starts to add expense to your protocol. Even more sensitive sort of technique is tyramide signal amplification. Again, this is a sort of multi-step technique where, again, you bind your primary antibody to your target protein, you come in with a secondary, and the secondary has to be horseradish peroxidase conjugated. Now, the secondary can either be a straightforward normal secondary antibody with an HRP conjugate, or it could be a HRP-based polymer system, which I'll explain slightly later. But what happens is the HRP catalyzes the deposition of, in this case, a biotin-related tyramide reagent, and then that can be detected using a fluorescent labeled streptavidin or an enzyme labeled streptavidin. And again, what you're getting is lots and lots of sort of amplification of signal. You're getting a lot of enzyme there, or you're getting a lot of fluorochrome there, so you get a nice bright, intense signal, so super sensitive. It's also quite unique in that the actual tyramide reagent binds to the tissue in really close proximity to the site of the antibody binding. It doesn't actually bind directly with the antibody. And that's a really important note, and it'll become apparent in a few slides why this is makes this reagent so unique. Now, about 5-10 years ago, lots of people were doing immunohistochemistry, double and triple immunofluorescent or double or triple detections were a bit more specialised. Now they're becoming very mainstream, and there's lots of different approaches that you can use to detect 2 or more proteins in the tissue. So, in this example, you can do simultaneous detection. So if, for example, you had 2 proteins that you're interested in and you've got an anti-mouse antibody against one and an anti-rabbit against the other, and you're happy that they're not going to cross-react with each other, they can be applied at the same time to bind the respective target proteins. And then in a second step, they can be detected using appropriate second reagents. So you'd have an anti-mouse reagent and an anti-rabbit reagent. And again, kind of an effective technique if things are kind of well expressed, good for sort of colocalization studies. But you've got to be absolutely sure that what you're seeing is true colocalization and not cross-reaction as a result of these antibodies cross-reacting non-specifically with either something in the tissue or with each other. Another approach is to do a sequential detection where you'd add your primary and then the appropriate secondary for that, and then repeat that process. And this can be repeated several times. So if you have appropriately selected reagents, you could repeat this process 3-4 times perhaps. Now, this is probably the ideal result if you're doing sort of multiple detections. And what we've done is we've specifically chosen antibodies and protein targets where we were confident that it wouldn't be expressed in the same area or the same part of the cell. And you can see from the composite image that we have 3 fluorescent signals here, and they're not colocalizing with each other, they're not cross-reacting. So it tells us that that's probably a true pattern of expression in that particular tissue, and that these reagents are not cross-reacting with each other in any way. And I'm sort of harping on about cross-reaction, and this is the reason why really. So, if you look at this specification sheet for a fairly standard secondary antibody, so it's a rabbit anti-rat biotin-related reagent, you would be using that and expecting it to bind anti-rat or rat primary antibodies. But you can see here they've given us some good information about it, and we can see that we have a 12% cross-reactivity with hamster, horse and mice. Now that would certainly be enough to give you a strong signal, that degree of cross-reactivity. So if you're using this reagent on hamster tissue or horse or mice tissue, for example, you may expect a background level, or if antibody is raised in these species or in part of your detection system, this antibody would probably bind to them. So the cross-reactions could be 2 types. You could have the situation where you may have, say, a mouse primary antibody, and you visualize that, and say, in this instance, with a red secondary, which is an anti-mouse, and then for your other antibody, which may be rabbit, your anti-mouse cross-reacts with it. Okay, so you get a false positive signal. Another possibility for these cross-reactions is it's not your secondary antibodies cross-reacting and binding to the primary antibodies, but they bind to each other. And again, that could give you a false positive colocalization signal. And if you are in that situation, it's quite difficult to say, this looks like a fantastic result. It could be true colocalization. But you've got to be absolutely sure that you're doing appropriate controls to make sure that that is the case, and it's not because your reagents have been purely selected and are cross-reacting either with each other or with the two different primaries. It's clear cut in this situation because there's no colocalization, therefore there's unlikely to have been any cross-reactions having occurred. So I'll jump back to sort of immunohistochemistry, and this is our sort of current method of choice in the lab, just because of the nature of the work that we do. Now again, I'll stress this is probably may not be perfect for everyone, but it does suit our needs. It's a multi-step process, so you normally do a peroxide block to block any endogenous peroxidases in the tissue, because we're using a peroxidase detection system here. There'll be a primary antibody step, of course. There's quite often a pre-polymer or a linking reagent. So in this example of one of the proprietary kits, we use that as a rabbit anti-mouse reagent. And then the polymer is a goat anti-rabbit polymer, which is an HRP conjugate. You then visualize that, and we usually use human toxin as our counter stream. Now, polymers are becoming increasingly available for a wider range of species. Certainly the earlier ones were aimed probably primarily at the diagnostic market, where rabbit and mouse primary antibody reagents predominate. But they are nice and flexible, and the protocol can be adapted for any species of primary by using appropriate linking or pre-polymer antibodies. And increasingly, HRP, or horseradish peroxidase, and alkaline phosphatase conjugated polymers are now available. To date, I am not aware of any polymers which are fluorescently conjugated. Now, if anyone in the audience has heard of any or knows if these are now available, I would be really kind of delighted to hear about them. So the polymer method, again, is a multi-step technique where you would use your primary antibody. In this example, it could be raised in mouse, or it could be raised in rabbit. You put on your pre-polymer, and then come in for your polymer system, which you then visualize with that. Now, you get the sensitivity from this method because of the large amount of enzyme that can be conjugated to this polymer backbone, which is bound to your detecting reagent, your detecting antibody. As I say, you can adapt these methods for sort of any species, really, as long as you know what the reagents in the polymer kit are raised against. Okay, so you could put any species primary, as long as your pre-polymer, for example, was a rabbit into any species. So let's say it's guinea pig, for example. So your primary would be guinea pig, your pre-polymer would be rabbit anti-guinea pig, and then your polymer would be a goat anti-rabbit. So that would bind to the kind of pre-linker and give you and visualize your antibody binding. Some of the polymer protocols now are just two-step ones where you can miss out the pre-polymer. So it's just basically you've got a rabbit primary, for example, and then a polymer which is a goat anti-rabbit HRP. Now, I think what had happened is the initial kits, they were designed primarily for, as I say, the diagnostic market, where rabbit and mouse ones predominated. So what the manufacturers were doing is they wanted to sell one kit that was suitable for both mouse and rabbit. So hence this sort of linking reagent, which made it compatible with both mouse or rabbit primaries. But if you understand what your kit is made of, you can certainly kind of adapt these to your specific needs. And what we were hoping for really is, nice clean staining specific with kind of low background levels. So the advantages for us really with polymers is that they are nice and sensitive, and they remove any issue with endogenous bacterial problems, which ultimately will save you time if you're not having to do endogenous abdomen vacuum blocking steps, for example, and potentially a bit of money. The disadvantages is that they may not be suitable for every antibody. You really got to sort of determine that empirically. Certainly in the early days of polymers, they were quite bulky molecules, and for some target proteins, there was also some sort of steric hindrance which prevented them from binding, as well as perhaps a normal streptavidin detection system. However, I think the polymer technology is advancing all the time. They're becoming much less bulky, and certainly in recent years, we've seen that as being much, much less of an issue. And with the polymers, as I say, they tend to be enzyme-based rather than fluorescent, but you can still do kind of multiple target detection. There's lots of different combinations of antibody, enzyme, substrate out there. And this is a particular combination we like, where we visualised in this sample smooth muscle actin in brown using DAB polymer, and then these Leydig cells with the sort of bluey purple substrate. That's actually just a fast blue substrate using the phos detection again. And there's sufficient contrast between the fast blue of the substrate and the Harris's hematoxin that we use. And you can see quite clearly where you've got expression of protein compared to counterstrain. As I say, there's lots of different permutations out there that you can use, and there's some sort of developments in sort of camera technology now where using spectral mixing you can have colours which are very closely overlapping, which can be separated to give you individual signals, but that's kind of moving in the realms of most advanced imaging techniques. And really, I think what we're aiming for at the end of the day, certainly in our environment, is a sort of panel of techniques that we can apply to a diverse range of samples and research questions. So whether it be formalin fixed paraffin embedded tissue, in the sample at the top, it could be cultured cells fixed in acetone methanol, for example. Could be cryostat sections or free-floating frozen sections, or it could be kind of whole-milk preparations such as the blastocyst in the bottom right-hand corner, or kind of small zebrafish preparations, etc. So a kind of common misconception that we have, well, not that we have, but that we encounter, is that a lot of people think that if you're doing a double or a triple stain, that all your primary antibodies have to be raised in a different species. Well, that's not the case anymore. So it is possible to do same species primaries if you use Fab secondaries and other types of techniques that are out there. So the way this would work would be if you had a mouse primary, for example, for your target protein 1, you bind that to your tissue, you come in with your Fab fragment secondaries, and they will bind to your target. And in this case, we're using a sort of biotinylate Fab secondary, and then visualizing that using a fluorescent streptavidin. And because these are Fab antibodies, they only have one antigen sort of recognition site, so they can't then cross-react with other reagents in the detection system very easily. We routinely would use an AB block between detections because this is a streptavidin-based system. And then we would also do a block with a unconjugated, unlabeled Fab reagent. So that would just really mop up any free sites which were available on your primary antibody that the Fab secondary could bind to. You can then come in with your subsequent detection. You apply your second mouse primary, for example, and then visualize that in a similar manner. And we found that this is quite an effective approach, particularly if you're using monoclonal targets. So if you're two primaries or raised in mice that are mice monoclonals, then we find that this technique works quite well. Hasn't worked quite as well in our hands for polyclonal targets. So if your two antibodies were rabbit polyclonals, for example, we didn't have quite as much success. We still felt that there was a degree of cross-reactivity of the reagents, so it wasn't maybe ideal. And another way that people can do same species primaries is if you are using mouse primaries and they're of different isotypes, then you can get isotype-specific secondaries, and they will distinguish between one type of mouse primary antibody isotype and another one. So that's another potential approach that you could use for mouse antibodies as well. Now, for us, this is really a sort of breakthrough, sort of benchmark paper for us, and it really made the job of delivering sort of multi-antibody detections from the same species within our environment much, much easier. And it's a paper by Susanna Toth-Macy. So if you are thinking about tyramide approach and you want to use antibodies raised in the same species, you should certainly grab this paper. And the way this works is it combines tyramide signal amplification with an antibody elution technique. And the basic principle is this: you, again, apply your primary, detect it with an HRP conjugate secondary. So in this example, it's just a normal HRP secondary antibody, but you could use an HRP polymer as well, and we've done this successfully. You then apply your tyramide reagent, which is catalyzed by the HRP and deposits around the site of the antibody binding in really close proximity. The important thing here is that it's not actually bound to the antibody, which means that you can come in with a very mild epitope retrieval step. So it's a heat-induced epitope retrieval step where you can either boil the kind of stain section up in pH 6 citrate buffer for 2.5 minutes in a microwave, for example, or you can use the short antigen retrieval protocol and the staining robot. And what it does is it strips away the primary antibody and secondary that was used in the detection, but it leaves the tyramide behind. And then what you can then do is come in with your subsequent detections and build these up. And because you're stripping away your primary antibody and your secondary antibody, there's absolutely no chance of getting any non-specific cross-reagent from your reagents. This is just a quick example of some staining that we've done using staining robots, using this approach. And it's a very effective way to do kind of 3 or 4 colour detections with primary antibodies, which are either raised in the same species or raised in different species. And just another couple of examples here. And I think in these examples you can see that were prepared by Sean McPherson, you can see quite clearly that there's no co-localization of these proteins. Their markers have been selected specifically to mark individual cell populations, and you can see there's no merging or overlapping of colours, so we've got really good specificity here. And I think the other thing which is really good about tyramide as a reagent is that it doesn't seem to create a barrier for subsequent detections. You would maybe imagine that, if you're applying detection upon detection, that eventually you'll create this barrier that would prevent your antibody from gaining access to its target antigen in the tissue. But I think you can see from this one that doesn't appear to be the case. It's an important consideration if you're using colorimetric detections, because if you're using DAB, for example, and that was your first detection, it does form an impermeable barrier to subsequent detections. Okay, so if you're doing a colorimetric detection with DAB and alkaline phosphatase, for example, we would always do the alkaline phosphatase detection first, because the substrate colour reaction tends to be much more permeable than DAB, which is really impermeable. It's particularly important if you're looking at co-localization, because you don't want any physical barriers there. It will affect the results that you're getting. Perhaps less of an issue if you're targeting different structures or different parts of the cell. Now, this is a sort of ready reckoner that we have posted in the Walnut Lab, and it gives us a sort of indication of the sensitivity of different reagents, because we are often in a position where people come to us and say, okay, we've worked up this antibody and we use this detection, and what's at this dilution? Can you give us a fluorescent endpoint? So with a ready reckoner like this, we can quite quickly work out the best protocol to use this and what degree of sensitivity it will achieve. And we know this, we traditionally based that on something like a 1 in 100 dilution for a ABC HRP detection with DAB would probably work around about the 1 in 400, 500 range with tyramide. And also if you're using a direct system, which is much, much less sensitive, you're probably down at 1 in 10 or lower. Another approach that you could use, probably getting a bit more advanced and a bit more challenging. It's not something that we use because it doesn't have the flexibility for us working within a sort of service provider. For people working at a bench on a specific research project, this would be quite a suitable approach, where you use fat fragment complexes to label your primary antibodies. And the principle here is that you would bind your primary antibody with your Fab label secondary antibody in solution. Now, there will be surplus Fab secondary floating around in that solution. So what we then do is block that with an unconjugated Fab, which then removes any chance of that binding to your sample. You can then apply effectively your labeled primary to your tissue and then repeat that process. So you can do multiple detections in this way by binding your antibodies in solution first before you apply them to your tissue sections. As I say, it's not something that we use because perhaps it doesn't have quite the sensitivity that we need for a lot of our applications, and maybe not as flexible approach, it's maybe not as flexible approach as some of the other techniques that we use to deliver this immunostaining as a service. And I think the other thing to remember is that if you do start to think about your staining, and not just use a method that the guy in the lab next door uses because it works for him, you think about your particular application, your particular problem, and you start to understand the technologies and the sensitivities of the detection systems that they're using. You'll be well equipped to adapt customized protocols to meet your particular research needs, particularly if you're looking at sort of multiple targets. And in this one, they're using another kind of really valid approach where they're using the sort of sensitivity of different detection systems to facilitate the staining of 3 sort of target proteins. And the way it's done is that you apply your first primary at a really low dilution, and use a really sensitive detection which can visualize it, meaning that if you then have to use a second detection system which is much less sensitive, then that won't bind to your first detection and be visualized. So it's a way of kind of using the sensitivity of reagents and detection systems to get an appropriate result. So I'm not sure that was very well explained. We use robots extensively. We have to do it because of the throughput that we have to deal with. I mean, I think now they're used probably extensively in the diagnostic setting. Maybe not quite as common in the research setting yet, but I think it's probably coming. And they allow us to do kind of high throughput on lots of different protocols using lots of different detection systems for our customers. Now, like people, robots aren't infallible, and this is just a slide to just really make you aware of some issues that may crop up when you're using robots. So what we have here is an array of 60 slides which have been stained on a pair of robots at exactly the same time, using the same conditions. And I think you can see at first glance that it looks quite good. It looks quite reproducible. The staining looks even, consistent, and looks okay. But when you start to dive in a bit closer, you'll notice that one or two of the slides, I think hopefully you can see the cursor here on the screen. There's an area here which is transparent, so this tissue has obviously not had a reagent applied to it, and also there's another one down here on the right. Now that probably occurs because a lot of these robots are using capillary systems now, or certainly the one we use uses a capillary system, and if you get an air bubble in the system, or perhaps a piece of tissue gets dislodged from a section, it can form a bit of a barrier which prevents some sort of even flow of reagents. So occasionally you will get areas on robots which are not stained, and I think from our perspective that just seems to be a fact of life. It doesn't happen very often, but it does happen occasionally, and it's something you should be aware of. And you can really mitigate that issue by careful placement of sections on your slides, and make sure they're nice in the middle, they're nice and evenly placed, and they're within the sort of parameters of the slide, the cursor combination, whatever. Okay. If you're working with really small bits of tissue, this could be an issue because you can imagine that small bubbles could appear in the system and prevent certain samples from being stained. So could maybe 2 or 3 sections on a slide would maybe help mitigate this, or perhaps if you want to be really extreme, you can maybe have duplicate slides in the run as well. Truly important. Now I think it's kind of fair to say that there's a huge range of reagents out there for kind of modern researchers working with antibodies, which means there's a huge range that you can specifically choose for your research applications. And I think if you are choosing secondary reagents in particular, just make sure that they're really kind of specific, they're really highly cross-absorbed, and think about if they're likely to cross-react with your target tissue, for example, or if they're likely to cross-react with other antibodies that you think you're likely to use. Okay, it's a massive job, and it can be quite challenging. But if you do want the best results, I think you've really got to try and understand the technologies that you're using, and not just do, as I say, the same technique that the guy on the bench next to you does, because his application could be completely different. It could be using different tissue, different species, different fixation. I think one thing is fairly certain, certainly our experience has been that people were always wanting more. 10 years ago, people would be happy with one colour. 5 years ago, they'd be happy with 2. Now people are wanting 4, 5, 6 colour combinations and beyond. And also the ability to combine things like immunohistochemistry and immunoflurescence with other molecular probing techniques such as in situ hybridization or an RNA scope, and that seems to be the future. So again, as you're starting to think about your immunostaining strategy just now, try and make it future-proof as well. So I think that's me. Again, I'll just thank my teams, Mel, Ruth, Debbie, Laura, Robin, Gary, and Mariana, and especially Sheila, who no longer works with me, but was a kind of close colleague for 10 years or so. So I'm quite happy to take any questions now, if anyone has any. Thank you.
Thanks, Mike. That was a great presentation. I'm sure audience found it very useful as well. A couple of questions for you. The first one here is for staining muscle sections with oil red O. Are there adverse effects of freezing the muscle section on the slide before fixation and staining?
Muscle is, I think, a fairly challenging and specialised tissue, and it's not something that I've got huge experience with, especially frozen sections. But I think the point would be that I think generally what people do is they try and relax the muscle by teasing it out, and then you want to kind of snap freeze that as quickly as possible. Okay, so I would probably recommend that you use something like a supercooled isopentane bath, so supercool isopentane and liquid nitrogen, and then freeze your tissue in a mold in that isopentane. What the isopentane does is it kind of protects the tissue from sort of air bubble formation. So if you drop the tissue straight into liquid nitrogen, you get lots of bubble formation around the tissue. That prevents the penetration of the cold. If you use isopentane, it kind of prevents that. So that's probably the approach that we would take. You want to freeze it as quick as possible so that you don't get an ice crystal back on. I'm assuming that because you're saying you want to stain oil, it will be frozen sections you'll be using, because if you are using paraffin sections, the fat and the lipid that you're trying to detect oil will have long gone because it'll be extracted in the ethanol and sadness as part of that processing. So I think oil red oil for fat or muscle, it's got to be frozen. You've got to freeze it really quickly.
Second question, actually, I think it's from the same person actually, and they've read a paper which there was an immunobuffer used, and they were wondering what the benefits of this would be as opposed to using PBS. Now, the immunobuffer is it's a BSA saponin glycine sodium azide and PBS buffer, and they were wondering if there's any benefits to using an immunobuffer as opposed to just PBS.
This is still for oil red oil staining, is it?
I think it must be, yeah. I think it's from the same person, so...
Yeah, I mean, I think for, it seems over the top. You know, oil red oil is a colorimetric detection. It's based on the properties of the dye dissolving in the lipid. I can't imagine that the buffer would have a huge influence. So an immuno-based buffer for that, I don't think there would be any advantages really. You'd probably be, as I say, freezing the tissue, fixing it in some way, maybe buffer formaldehyde, and then staining it in your oil red oil solution, which has some isopentane in it, I think.
Okay.
So yeah, I don't think you get a huge advantage from that, but it's something I've never done. Okay.
Next question is why is there a preference for immunofluorescence rather than non-fluorescent immunos?
I think it's really, in our setting, is because of really the sort of advances in imaging technology. So there's really good fluorescent microscopes out there. There's really good confocal microscopes out there, and you can detect 3, 4, 5 channels of 3, 4, 5 colours at the same time using fluorescence. The other thing about these detection system, sort of microscopes is you can switch channels on and off, so you can look at 1, or you can look at 3, or you can look at 5, and you can look at them in different combinations. The problem with colorimetric detection is that if they do colocalize to the same area or the same cell, then the colours tend to kind of mix like paint. So if you've got a child and you start with red paint and green paint and blue paint and you mix them all together and get sludge, then that's what it starts to look like. So it can be quite difficult to work out exactly what's happening.
Yeah.
So I think for us that's why we use fluorescence, because it is so clean, so specific. You can switch channels on and off, so you can see things really clearly. Colorimetric is much harder. Now, there is, I believe, Kirk and Elmer have brought out a new sort of spectral imaging bright field camera, which can separate these colours. So, if you took your red, green, and blue colorimetric and you mixed it into mud, then this camera and that technology can separate that. But there's probably only a handful of people who have that technology. For most people, it's called bright field microscopes. And it's a good approach. I mean, we still use it, and it's a great approach for image analysis or doing point counting in a microscope, because it is easier. But I think you've got to have quite good separation of your colours, so therefore, not colocalization.
Another question's asked. A person had missed it. They said you'd mentioned alkaline phosphate, and they missed that part. What do you use alkaline phosphate for?
Yeah, so you can buy either secondary antibodies or polymer systems which have alkaline phosphatase as the enzyme that's conjugated to it. It was extensively used for in situ because the combination of alkaline phosphatase and its substrate, which is nitroblue tetrazoline and X-phosphate, is a really sensitive combination, because you get a really good sort of colour reaction that will develop over many hours. So it's nice and sensitive. Also, if you're using a DAP detection for your first detection app, you may want to use a different enzyme so you get contrasting, but choices of lots of other coloured endpoints. So HRP and APPAP, good combination, use it quite regularly.
I've got a final question. What, in your opinion, is the best controls to use?
Well, I think there's lots of different opinions of what controls you do use. I mean, we routinely would emit a primary antibody. So that's a fairly standard and fairly easy control. But all that control is showing you is what background the detection is going to. A more common control is to replace your primary antibody with a non-immune IgG of the same species. So if you've got a rabbit primary against smooth lactin, for example, your negative control might be a rabbit IgG. Okay. Now, the issue with IgG, certainly in our hands, seems to be that they quite often give you quite a high background naturally. It's a pooled immunoglobulin. You don't know what the immunoreactivity of the animal was that it was collected from. They're probably usually much higher concentrations, so you've got balances of concentrations. So quite often, you will still see a background level to use these as negatives. Another two alternatives which I prefer, another three alternatives, is peptide absorption. So some primary antibodies you can buy the peptide that it's raised against, or the protein it's raised against. You can cross-absorb that out where you're back to your tissue. So that's saying, okay, the antibody's been mopped up by the peptide, therefore it's not binding to the tissue, so it's real. You could use a primary antibody from the same species that's not expressed in your tissue, which would be another bad control. So that's saying that the IgG is just not binding, is not binding on specifically, and you've not got a background from the detection system. And the final one, which I'm starting to quite like using, is, particularly if you're using human tissue, is you use an antibody against green fluorescent protein. So unless you see humans walking down the street that go green, using GFP, which is not normally expressed, is quite a good control. And the advantage of that is these reagents have probably been, I think, purified in a similar way to your target primary. So again, I think that's another very good control. So I mean, there's lots of controls you can do. I think you've just got to, take your choice. But we definitely want to do something. And I think, especially if you're doing sort of localization methods, you've got to be quite careful with your controls and check that reagents aren't cross-reacting. And tyramide gets around that basically for us. We don't have to do extensive controls because we know it works and we don't get any crossover.
Thank you, Mike. So the end of the questions brings us to the end of the seminar. So thanks again, Mike, for an excellent presentation. And last but by no means least, a big thank you to you, our audience, for taking the time to attend today. If you have enjoyed the seminar and would like to view the video recording of the session, please visit the seminars page on bitesizebio.com. It should be available within the next 24 hours. There you can also check out our upcoming webinars. So, until next time, good luck in your research, and goodbye from all of us at Bitesize Bio.